Hi students, welcome back to the third part of the chapter Cell, the unit of life. So we have discussed a lot of cell organelles. Now we are going to discuss about plastids. Plastids are the unique feature of plants and as well as euglenoids. You may be knowing about euglenoids, euglena. So the organism who can swim like an animal but do photosynthesis like a plant. So they are also containing the plastids and plants. So plants having basically three types of plastids. You know that we can see from that image itself. Leucoplast for storage, chloroplast for photosynthesis, chromoplast which gives specific attractive colors to the plant cell organelles. Now let us see the type of plastids and their functions. So basing on the type of pigment contains, we have different type of plastids. First let us discuss chromoplast. So chromoplast are the colored plastids which give attractive colors to the petals and fruits. So you can see the cells filled with the chromoplast. Then it appears like this to the petals of flowers to attract the pollinators, and also it gives a very attractive color to the fruits to attract the animals for dispersal of seeds. So that is a basic function of chromoplast, the attraction, give bright colors. Okay, the next one is chloroplast. In the case of chloroplast, it has double membrane bounded structure. Okay, so that is a special feature of, again, eukaryotic special feature, double membrane bounded organelle, and that is a main characteristics of plant. Why do plant become the chief producers of our biome or biosphere? Because of they are having this pigment inside the chloroplast. The name of the pigment is chlorophyll. So you can see that it is basically present more in the leaves. In the leaves also, the mesophyll cells are having more number of chloroplast. It varies from 1 to 20 to 40. Suppose if it is saying about Chlamydomonas. Chlamydomonas is a algae which is having the disc shaped chloroplast. We had seen Spirogyra, spiral shaped chloroplast, oval shaped, round shaped, various shapes are there in chloroplast in the plant world. But in the angiospermic plant, it's normally oval in shape. The number will be large, depend on the type of cell. Basically in the leaf, their number will be very high per plant cell. So if you see the inside the chloroplast, inside the double membranous structure, the space inside is called as stroma. The stroma containing matrix. So you can see the stroma over here. Excuse me. So you can see the stroma over here. So you can see the stroma over here. So stroma is present inside the membranous structures. It also having the chloroplast DNA and granum. The stacks of thylakoids called as granum. External internal membrane is here. Then intermembrane space, the space in between the membrane, and structures like plastoglobules, ribosomes, etc. are there. Thylakoid is the structure unit of grana. It having outer intermembrane. The lumen of thylakoid is very important, especially for photosynthesis. The pigment is present in the thylakoid membrane. And the lamella, stroma lamella, which connect the thylakoids of grana. So this is all the part of that. Okay. So the grana having the unit of flattened sacs, that is called as thylakoids, which contain in the chlorophyll pigment, which have the ability to trap the sunlight for photosynthesis. So see, we can see here a part of thylakoid membrane is cut open and shown here. It's having different electron transport systems. This all we will discuss in the chapter photosynthesis. Okay, in plant physiology. So the thylakoid membrane is there, which helps, which having chlorophyll, which will help for the trapping of sunlight. Okay, just think about that. In a tree, a big tree, hundreds of leaves. In a leaf, cross section of the leaves, upper part, palisade, spongy parenchyma. These are mesophyll cells. In the mesophyll cells, there will be 30 or 40 chloroplast. In the chloroplast, around 10 grana. In a grana, around 10 thylakoids. Thylakoid membrane, outer membrane, we are having this chlorophyll where the light is trapped to produce the energy, where the light is used to split the water into hydrogen and oxygen. So what a fantastic technology has made to do the photosynthesis. Only plants can do that. Okay, so that is the structure peculiarity of chloroplast. And already I told you the stroma lamella is here, the flat structures which connect the thylakoids of one grana to the thylakoids of the other grana. So you can see here the grana structures. In that, the single one is a thylakoid, and that connect with one thylakoid to other one, that is the stroma lamella structures. Okay. And now let us see the other type of plastid, that is called as leucoplast. Leucoplast, initially we told that is a storage plastid, that is colorless. So mainly it is present in the storage tissues. So which is our storage tissues of plants? That is parenchyma. It can also see in the pith regions, etc., where the food is stored. So there are different type of leucoplast basing on what type of material they are storing. Some of them will store starch, some will store protein, some will store oil particles. So depend on the things, just like see the sunflower seed that store oil, sunflower oil is there. Groundnut, we put a groundnut and break it and rub it on a white paper. We can see a oily tint. But you see rice, wheat, potato, carrot, beetroot, in this all these things we can see that we will store starch. So a different type of things will be stored. So the names are like that. Amyloplast is a leucoplast which store carbohydrate. Elaioplast that store oils. Eleroplast that store proteins. So in different type of cells, different types of or different type of area, different type of leucoplast are there. It store food in different form. Okay. Then that is about the three plastids. So now we are seeing chromoplast, chloroplast, and leucoplast. The next one are ribosomes. We told initially about ribosomes in the prokaryotes. A ribosome is made up of RNA and proteins. Ribosomal protein or ribosomal RNA. Again, that we will discuss in detail in class 12 about ribosome, structure of ribosome, ribosomal proteins. Okay, different type of proteins, etc. In the case of this is what we are telling about eukaryotic ribosomes. They are 80S. Again, yes, I told you the Sedberg coefficient, or it is a Sedberg unit. It is a, what is that? Density centrifugation coefficient. That is what, basing on that 80S, and the subunits are 60 plus 40. Again, I told you it's not mathematics. 60 plus 40 subunits joined together, it is 80S in eukaryotic ribosomes. Their main function is to produce protein or a site of protein synthesis. You can see here the mRNA, which is coming from the or produced by the RNA, okay, by transcription, and on the mRNA the ribosome will read the message. That's a mRNA, messenger RNA. The ribosome can read the message, means on what basis it is written. So basing on the letters on mRNA, basing on the nitrogen base on mRNA, basing on the nucleotides on mRNA, ribosome will bring or attaches the amino acids. So basing on the message of on mRNA, ribosome can produce the required protein. So once the protein synthesis is over, ribosome again become dissociate into larger and smaller subunit. Whenever protein synthesis is required, that time only that is smaller subunit and larger subunit will unite along with the mRNA for protein synthesis. So that is about ribosomes, and previously already we told about polyribosomes, the structures what we are seeing in the case of prokaryote. Okay, so in a single mRNA we can see numerous ribosomes working at a time. Okay, that is polyribosomes. Now let us discuss about microbodies. Microbodies are a special type of organelles present in almost every type of organisms, whether it is plant, protozoa or animals. Okay, so examples are peroxisomes. You may have not heard about them because it is less discussed. So they have some enzymes to the breakdown of fat, alcohols and amino acids. Why they have to break down fat, alcohol and amino acids? Because they have special functions. So they have special functions like detoxification of peroxides, hydrogen peroxide you heard about. Okay, so it is there in the case of, here you can see glyoxysomes. What they will do, fatty acids is utilized in the production or in the synthesis of glucose. Okay, so this is what I told you, the energy producing mechanisms, and as well as in all these areas they functions. So peroxisomes are there, glyoxysomes are there. They are mainly working on detoxification of materials or chemicals, alcohols, etc. So we know liver will be having more microbodies. Liver will be having more peroxisomes because liver has a function of detoxification of poisonous material and alcohol in human body. It also take part in photorespiration in plants. We will discuss that in the chapter of plant physiology. Okay. So now let us discuss about the cytoskeleton. Cytoskeleton are the network of filaments, especially proteinaceous filaments, that gives the perfect shape for the cells. Excuse me. So it containing microtubules and microfilaments. So like that of, we can see actin, myosin. So actin, myosin, these and all actin filaments, especially these all are proteinaceous filaments. So these proteinaceous filaments will give the network-like structures which give the perfect or proper shape. So what we are seeing, the image of plants or animals are normally shown without the cytoskeleton to get a clear image of organelles. So actually cytoskeleton is here, and endoplasmic reticulum also contribute in the formation of cytoskeleton, and here the proteinaceous filaments helping for the shape. So it also gives the mechanical support. It also helps in motility, maintain shape, just like see that movement of amoeba, a movement of WBC. So that type of motility also, for cytoplasmic streaming, along with that the changes in the cytoskeleton is also required. So that is what is the major function of the cytoskeleton. Now let us go into the structures called cilia and flagella. You have heard about cilia and flagella. Cilia is present in the microorganisms like paramecium, okay, and flagella we can see in the bacteria, euglena, etc. And the same cilia is present in our windpipe, trachea, to propel out the sputum or the mucus trapped with microbes. And also the cilia is there in oviduct of female reproductive tract to transport the ova or zygote. But flagella, already we have told about that flagella is longer, cilia are smaller in structures. Here the flagella is shown with a prokaryote, cilia is shown with a eukaryote, somewhat like that. So here you can see the paramecium is moving or propelling with the help of its cilia. So you can see the oar, like an oar they are using, just like how do you swim. How do you swim? In that manner they are using the cilia, the swimming manner. As you can see like that, how do they use the cilia? Cilia, how do they swimming? Backward stroke and forward stroke. That how do they swim? But how do the flagella is using? Flagella is just whip-like movement, just like it move like that. So with that help of that, the organism can move. So whatever it may be, cilia and flagella are locomotory organs, may be present in different type of organisms. Okay, so we can list out the name of the organism which present which containing cilia and flagella, and in human body also where it is present, its role. Okay, so next we can see that the internal structure of the cilia and flagella both are having a similar internal anatomy. We are not going to study the detail about the cilia and flagella ultrastructure. They are having a plasma membrane covering is here, and the core is called as axoneme. Axoneme is with, you can see the core axoneme, and it is having a 9+2 arrangement of microtubules. What is 9+2? This is 2. 2 are there in the middle, and the 9 are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. So that is what is called as, it's a cartwheel arrangement. Cartwheel. Bullock cart you have seen. Anybody have seen bullock cart? Bullock cart wheel. How does it look like? So cartwheel arrangement or cycle wheel. Cycle wheel, how does it look like? The spokes, central part, and the spokes radiating into a different part. In that manner it is present. So that is what is called the structural anatomy of cilia and as well as flagella. And the central tubules, you can see the both central tubules are connected by the bridges. Here you can see the bridge is present in the central tubule. Further, and there is a sheath is here which cover our central tubule, and it connect to the peripheral double X. So this is called as double X. But two are there. So the peripheral doublets are connected by radial spokes. Connected by radial spokes. So this is what the particular features of the cilia and as well as flagella. Now the centrosome and centrioles. So the centrosome and centrioles, you can see that centrioles are present in centrosome. Okay, so it is a very characteristic feature present only in the animal cells. Okay, it is surrounded by a pericentriolar material. You can see the pericentriolar material will be there around this. Okay, you can see here very small centrosomes. Okay, inside that this type of centrioles. Okay, so this is what the structure, and here the peculiarity is that 9 plus 0 arrangement. Of course, the radial spoke is there and cartwheel structure is there, but in the center there is no microtubular arrangement, but only in the periphery there are 9 triplets are there, and that is connected to the center. There is no separate by triplets or some other structures. Okay, and the function of the centrioles, of course I told you already, it is helping for the cell division. Okay, that is what I want to show you here. During the cell division they will use to connect with that of the spindle fibers. You can see the spindle fibers are originating from the centrioles that pulls the, we'll discuss in the cell division chapter in detail. Okay, it's like the chromosomes will be there. When it is pulled into both the side, it splits through kinetochore and move into opposite poles. And they move into opposite poles, that is because of the shortness of, or that they get the getting shortened. Which one? The spindle fibers, that is started from centrioles. So that is another function. I already told you the basal body of flagella and cilia. Cilia, flagella, just now we discussed it. Its basal body where it is connected with the, just like that. No? So the organism's body will be there, the flagella or cilia is connected as a basal body. Also who will work? The centrioles will work in the unicellular or smaller organisms. So these are all the basic function of the centrioles. The structure of centriole difference with that of cilia and flagella, where the spokes arrangement and as well as central cartwheel arrangement, that is difference. Okay, please note it down. Then let us go into the final cell organelle, that is nucleus. Why nucleus? And nucleus is the most important cell organelle in the plant or animal. You can see that center part in the animal cells. Okay, and it is also a double membrane structure. Previously we already told that eukaryote, prokaryote, the caryon means nucleus only. Okay, so basing on nucleus having the membrane, nucleus is present or not, basing on that double membrane structure is present or not, we are categorizing the organism into prokaryote and eukaryote. So double membrane is there, and in between the membrane a space is there. That space is called as perinuclear space. That space is called as perinuclear space. So that further inside the nucleus containing chromatin, and the chromatin, nuclear matrix is there, and nucleoli are there. Three basic components inside the nucleus. Nuclear membrane, that is a double membrane structure is there, outer inner membrane, in between perinuclear space is there, chromatin reticulum is there inside. Okay, and a matrix, the liquid or semi liquid component is there, that called as nuclear matrix or nucleoplasm, and nucleoli. Nucleoli is a round structure. You can see here nucleolus, you can see here, and you can see the nucleoplasm here, and the chromatin is here, chromatin reticulum. Okay, so this is about the nucleus structure. And the nucleoli, already we told it, the function is for the rRNA synthesis. What is the rRNA? R is small, then RNA is capital. So R for first R is for ribosomal RNA. RNA, DNA we know. What is RNA? Ribonucleic acid. Ribonucleic acid. rRNA means R is for ribosomal. That means already we told about that the ribosome is made up of RNA and protein. So this is rRNA. This rRNA is prepared by or made inside the nucleolus. Nucleolus function is to produce that. So where the cells have to produce more protein, so they should have more RNA. More RNA means more RNA required. That rRNA required means nucleolus number may be more or their size may be more. So that is what is very important. Then if you go to the structures of nuclear membrane, outer membrane is continuous with the endoplasmic reticulum. So you can see the endoplasmic reticulum, the reticular structure, network structures already we had discussed that. It is the outer membrane is continuous with the ER. Because as it continues with the ER, it having the ribosomes also. As the ribosomes are there, we already told it is related to protein synthesis. And the nuclear pore, the hole here, or you can see the gap in between the nuclear membranes, nuclear pore, helping for communicating with the cytoplasm. You can see here the perinuclear space. You can see here outer membrane, inner membrane, perinuclear space in between, and nuclear pore complex. You can see here helping to communicate outside. And in that, very importantly, we have to say that is a particular cell or type of tissue, or we can say the erythrocytes, commonly we call as RBC, of mammals don't have nucleus. So only one mammal which have nucleated RBC is camel. Okay, and the sieve tubes of vascular tissue. Where is sieve tubes? In the phloem tissue. Okay, in the phloem tissue of the vascular plants, or the plants, angiosperm, gymnosperm plants also does not have nucleus. This is a very peculiar feature of these two type of tissues. Now let us study about the other components inside the nucleus. Basically, we are going to the chromatin. So chromatin is DNA plus protein, basically non-histone proteins from the chromatin. Here you can see that it's not a bowl with noodles. Okay, it is shown the nucleus, and nucleus with the chromatin is there, thread-like structures. If you just stretch out the whole chromatin or whole DNA, stretch out, it will go up to 2 meters. It will go up to 2 meters long. So what is here? That chromatin means DNA with the histone and non-histone protein is wrapped, then make the chromatin. And the chromatins during the cell division, it condenses, compactly arranging, condenses into chromosomes. So this is what we can see, the condensed form of coiled and condensed form of chromatin as chromosomes. So again, nuclear pore communication, or the chromatin will go out in order for any, just not going out. Okay, the RNA will go out to the nuclear pore, into the nuclear plasm. Cytoplasm communication. Okay, so cell division time, during the cell division time, what will happen? The chromatin will coil, I told you, the coil and condense and form the chromosomes. Compact structure, chromosomes forming during cell divisions by coiling and condensation of chromatin reticulum. For coiling and condensation, they required different type of proteins that generally we call as histone protein and non-histone protein. That's for our level, that is enough. Even for 12th class also we are going to discuss that. Histone is a type of proteins, H1 protein is there, and these two will help to wrap the DNA over the histone proteins. So the DNA is wrapped over the histone proteins and form the nucleosome, and this will be coil and further coils and forms this compact structure, chromosomes. Okay, so you can see the chromosomes over here, in different times. So before cell division, this is the chromosome structure. Beginning, you can see this is the chromosome before cell division. This is the chromosome before cell division, ready for cell division. This is after cell division. This is after cell division. It looks like this. Okay. What is further about chromosomes? So you got chromosome, genetic material, the DNA. So remember, what is chromosome, what is chromatids, what is DNA, what is gene. 4 parts are there. So what is DNA? Double helical structure of DNA. We know, isn't it? So a part of DNA that code for maybe from here to here. We cannot say exactly. The part of the DNA which code for one protein ultimately, that is called as gene. Control one character, that is called as literally called as gene. Okay, so part of DNA. When DNA is wrapped over the histone proteins, little coiling, it become chromatid. When chromatid further coil and supercoil, it become chromosome. So that is what the structure. How does it goes? Chromosomes are the supercoiled structure of chromatin, DNA, etc. Okay, so that is what I am going to show you here. You can see the DNA is here wrapped around the histone proteins, that is called as nuclear DNA. Then again nucleosomes, again further coils, supercoils, and forms the chromosomes. Okay, in human body, in human being, every living cell, every cell, we are having 23 pair of chromosomes. 23 pair of chromosomes, except in our gametes that we know, they are having 23 chromosomes. Like sperm and egg, they are having 23 chromosomes, remaining all the body cells are 23 pair of chromosomes we are having. This is also going to study about the role of chromosomes in the coming chapters. Okay, cell division is there, cell division, cell cycle and cell division. There we will discuss that. Now let us see the part of chromosomes. You can see chromosomes having arms. This is called arms of chromosomes. Okay, that is called as chromatids. Arms of chromosomes called as chromatids. Okay, and where it is joining, that area is called as centromere. The area where it joined, that is called as centromere, and the centromere is having a disc-like structure. Okay, so join in the sense means primary constriction. So you can see that there is constrictions here, here, here. So where the primary constriction is there, that is called as centromere. The centromere is having a disc-shaped structure, that is called as kinetochore. Centromere is a primary constriction. Kinetochore is the area where we can see what is the role of kinetochore. You can see over here the spindle. I had shown you that about when you discuss about the centrioles and spindle fiber formation. The spindle fibers are attaching into the kinetochore region of the chromosomes. So that is the role of kinetochore. Centromere is a primary constriction. Okay. Now classification of chromosomes. Another very important area. Classification of chromosomes. Chromosomes are classified into 4 basic types. 4 basic type. But we have 23 chromosomes. I told you 4 basic type basing on the position of centromere. You can see the centromere position in this type and in this type, that is slightly differs. The first type is metacentric. Metacentric means chromatids are equal in size, almost equal in size. Okay, from both sides chromatids are equal in size. The primary constriction almost in the middle. Centromere is in the middle. That is called as metacentric. Okay, the classification is a commonly asking question. Okay, then what is submetacentric? Centromere is slightly away from the center towards one side. So in one side chromatids are longer, another side it is little shorter arms. So this is called as submetacentric type of chromosomes. Then you can see the third one is acrocentric. Acrocentric, the centromere further moves to the one side. Here the one side arm is very long, another side arm is very short. So it is acrocentric and telocentric. It is almost in the terminal side. We have centromere. Centromere is almost in the terminal side, that is telocentric. So on the basis of this arrangement or position of centromere, 4 types: metacentric, submetacentric, acrocentric and telocentric. Okay. Then one more thing we have to study about the chromosome, that is satellite. So what is satellite in chromosomes? So it's a secondary construction of the chromosomes, not in all chromosomes. So it appears like a small fragment. It is still united with the chromosomes, that is called as satellite. Okay. So the role of satellites we are going to discuss in the next year again. Next year only, because the role of satellites in DNA fingerprinting and role of satellites in identification of persons is all very important. Okay, so the non-stained portion of secondary construction of chromosomes, that is present only in few chromosomes, that is called as satellite. Okay, so this is all about this chapter, and your chapter has been completed. Thank you.